Hello everyone, welcome to today's session. Myself Pushpendu. I'm a biology subject matter expert from Toppr.com. So today in this session we are going to talk about a very interesting and also a very important topic. First of all, in terms of your examinations. So this will cover both the boards as well as your medical entrance examinations. Okay. So what's the topic? The topic will be biotechnology. We'll be talking about some of the important tips related to biotechnology. So how are we going about this session? We'll be going to cover in this session about some important concepts that you must know about this topic biotechnology. Okay, so we'll be talking about some of the important concepts in biotechnology, and those are actually the concepts which you will require and which are the favorite areas at the examiner's point of view. Okay, we are also going to talk about some of the frequently asked questions, the areas where the questions are frequently asked, and by the time I'm continuing with the session, please post your doubts. So in between the sessions also, and lastly also, we'll be talking about and discussing about your doubts. Okay, so first of all, why we have chosen this topic biotechnology. So you must be aware, right, that bio, it consists of various terminologies, too much of names, too much of learning, remembering, right? But this is the area which contains about 10% of the weightage. So you can surely expect about 3 to 4 questions in your examination, okay, in your entrance examination. So you can actually secure about 12 to 16 marks easily from this topic. So the importance of this topic is that you do not have to learn or mug up a lot of terminologies over here. If you're clear with your concepts, then all it's done. So you can easily secure about 12 to 16 marks and add to your score. Okay, so let's get started quickly. I'm sure you will enjoy this session. First of all, what is biotechnology? Okay, one important tip about biology is that we must be clear about the terminologies, right? Because if you're clear about the terminologies, you will be at least 30% clear about the concept, and the rest will be then with the theories and all, right? So if you're clear about the term, then you can actually know why are we going to study about that, what is the basic concept behind this. So let's try it. Biotechnology. Okay, if we break the term, what it's coming like bio and technology. Technology, it's a general term. So this actually involves various techniques using live organisms, specifically the plants, animals, their cells, their components. Also very importantly, microorganisms. Okay, so we have used these type of live organisms to produce various products which are useful for us, okay, which are useful for human beings. Now, this process of biotechnology, it involves various techniques, very important and interesting techniques. What are they? They are like DNA manipulation, recombinant DNA technology, and genetic engineering. So we're talking about these concepts soon. Now, another important tip about biotechnology, about biology and their entrance examinations is the examiners. So you can easily expect the questions which ask the names of scientists. Okay. So this biotechnology term was termed by Carl Urke. Now it's one of the favorite areas for the examiner's point of view. So there's no need to panic, because you understand there are about 38 chapters according to NCERT, and there will be a huge number of scientists. So how to go about it? Okay. So what I would suggest you to follow is, like, you must try to understand or remember those names of the scientists which involves those important concepts. So I'll try to explain this as we go through this session today. So you'll see that what are the areas or which are the specific scientists. The probability of asking the names of those scientists is more for your examinations. Okay, we'll learn this. So there's nothing to panic about it. So this topic of biotechnology, it involves two main techniques. What are they? First of all, try to understand genetic engineering and chemical engineering. So we are talking about genetic engineering, right? Genetic engineering, it involves around manipulating the genetic material. That means, so we try to change the wild type genes of a particular organism, correct? So this involves the genetic engineering. Now, why chemical engineering? Now, it is just not enough that if we have manipulated a gene or an organism, it's just not enough. We have manipulated something, but it required to be stable. Okay, we should maintain a sterile microbial environment and prevent their contamination, right? So otherwise it will not be of any use, correct? So we must actually do something. Chemical engineering is required so that we can help in the growth of the desired organisms, correct? Now let's talk about two important discoveries which actually I'll say that these two important discoveries actually helped for the advancement in biotechnology. What are these? First, the plasmids. I'm sure you must be aware of this term plasmids. So we'll quickly try to understand what are plasmids. Plasmids, these are extra chromosomal DNA. So please try to take a note on this term. It's extra chromosomal, okay? Now, why extra chromosomal? That means this is not a part of the genomic DNA, correct? That's the reason I'm referring it to as extra chromosomal DNA, correct? Now, the question is, very important thing you must understand, why does these type of organisms, specifically the prokaryotes, okay, the bacteria, they have these type of plasmids. Why do they need plasmids? Plasmids actually, they provide additional advantages to these type of organisms, correct? A very good example is like they provide antibiotic resistant genes. So these are carried in the plasmids. So basic concept about plasmid is that we have used plasmid in order to, we have actually used plasmids as a vector, okay? Suppose if I have to send something from one place to another, right? We'll need some carrier who can take that thing or that stuff from me and give it to the actual recipient, right? So what about the DNA? How can we do that? So we need vectors and plasmids. You won't believe, plasmids are the most common type of vectors which are used. This is the first one, right? Now, what's about the second one? Second one is restriction endonuclease, okay? Now, restriction endonucleases, these are very commonly termed as the molecular seizures. So this you must be very clear about restriction endonuclease because these are the enzymes which actually are capable of cutting the DNA molecule, cutting the genetic material, okay, whether it's RNA or DNA. So it can cut those DNA strands. Now this is very important because you understand, if we want to play around with the DNA molecules or the genome, then we must be able to cut it and paste it somewhere else, okay? Then only we can actually perform the process of biotechnology and various advancements. So restriction endonuclease is the second most important discovery in terms of biotechnology, okay? So again, we should remember important scientists over here. I have told you that we'll try to understand which are the scientists which you must understand and know the name. So this was first isolated, the restriction endonucleases, it was first isolated by W. Arbour, okay? You should remember this. Now, also there's a very common question which is being asked is, which is the first restriction endonuclease that was discovered? It's, remember the name, it's HIND. HIND2, okay, HIND2. So it is isolated from an organism called Haemophilus influenzae. Now we will be talking about the next type or topic, which is the GMO, which is genetically modified organism. So again, from the name we must try to understand, what did I say? Genetically modified organism. So we'll be playing around the genes, correct? The genome modified, we'll be modifying them, and we'll be of course getting some organisms which are genetically modified. So for any steps in biology, any process in biology, we have a series of steps, okay? So how to remember those steps? So I'll just I actually would suggest you to think very simple, okay? What would be the simplest way to achieve that goal, correct? Then you can easily get on to the steps or remember the steps. So here, for the creation of genetically modified organism, what should be done? So first, clear your goal. What is the aim? What is the concept behind creating this type of GMOs? So the concept is, if a particular organism lacks a particular type of gene or a particular type of product, then what should be done, correct? So is it possible that we are going to take it from any other organism and give it to this organism, suppose organism A, which actually lacks these type of characteristics? Yes, it is possible. So this is our target, okay? You understand this? So what should be the first type? The first thing it will be like, it is the identification of the DNA with the desirable gene. So we must first try to identify which are the particular type of genes which we desire for, okay? Which type of characteristics? What should be the second step? Now we have first we have isolated or first identified that thing. So we must isolate that, and then what should be done? Then we have to introduce that identified DNA into the host, correct? Host means the organism where we want that this particular type of character should be expressed. So it should be there. Now, the second thing, as we have already mentioned, that we have transformed the host. So almost we have created the GMOs. So we must be able to maintain the introduced DNA inside the host, or else, because there are many enzymes which will actually cleave and destroy those type of DNA, because you understand it's a foreign part. Okay, it's not a part of the normal host cells. So we have to maintain that, and this transfer of the DNA to its progeny. So it's not only enough that we have created a host, but it should be able to transfer to its progeny, then only it will be proliferated further. Correct? So these are the basic steps. First we identify, then we introduce it into a host, and then we try to maintain it, and so that it can be transferred to the DNA, okay, to its progeny. Correct? Now, for the biotechnology process, what are the various tools we are going to use? First, can you name it? The first thing which we use is enzymes, correct? So there are different types of enzymes. First of all, the lysing enzymes. Why? Suppose you have identified a bacteria which has certain characteristics, correct? So you have to isolate it, okay? So you have to lyse that bacterial cell. Cell wall of the bacteria should be lysed. So this involves enzymes, which the lysing enzymes, correct? For example, in plant cells, we have to use cellulase. For fungi, we use chitinases. So these are the lysing enzymes in order to break open the cells, correct? So we have talked about the lysing enzymes. Now, the next enzyme which we use is the cleaving enzymes, okay, which will chop off the DNA. Now these are of various types, okay. This is very important. You should remember about this, and you can expect questions regarding this. So what are this? So they are the exonucleases. That means exo, correct? So from outside. So these type of nucleases, they cleave from the outside areas, okay, from the ends of the DNA molecule. What about endonucleases? Correct? So exo we have covered. What do we mean by endonuclease? Endonuclease will be cutting from inside. Now there is a special type which is called the restriction endonuclease. So you understand, I have talked about endonuclease, and again I am talking about restriction endonuclease. So there must of course be certain differences between these two. Now if you talk about endonuclease, this is very important. Actually this can be asked in terms of your board examinations. So if you talk about the endonuclease, it cuts only at single strands. But if you talk about the restriction endonuclease, it cuts at the double helix, okay, the duplex DNA. Both of the strands of the DNA, they are actually cut, correct? So this is the important difference between these two type of enzymes, the endonuclease and the restriction endonuclease. Again, I will be talking about a very important area which is very probable to be asked in your examinations, okay, the naming or the nomenclature of the enzymes. We'll take one example, that is EcoR1, okay, ECOR1. Now, EcoR1, this has been named from, because the enzymes has been isolated from an organism, Escherichia coli, okay, E. coli, you must be aware of this. From there, you understand the letter or alphabet E has been taken, then CO, Escherichia coli, and the strain name is R, and this was the first restriction enzyme which was isolated. So it has been given the number 1. So this is the way the nomenclature for the enzymes is done. Now this enzyme, it has a specific DNA site, okay. Now the very important thing about restriction endonuclease is that they are very specific to its sites, okay. They can cut at specific DNA sites only. Now what is that site? That is, you must remember, it can also be asked in your examinations, that is GAATTC, GAATTC. And where will it cut? It will cut between the G and A, okay, for very important both of the strands, correct? Now this is the area where you can again be asked questions. So you can be given a name of an enzyme, and then you can, and in the options you may be asked about the different organisms. Now you have to choose the correct option from which that organism or from which that enzyme has been isolated, okay. So I hope you have understood how this nomenclature is being done. Now, a very common question which has been asked most of the examinations, medical examinations, is where there is a tricky question which asks you about, which wants you to understand or answer about ligase, the polymerases, the reverse transcriptase, and the restriction endonucleases. So you must be properly able to differentiate between all these 4 types of enzymes I have named. So let's quickly understand what are these. Ligase, okay, that means they are able to ligate, they are able to join. What do we mean by polymerase? Correct? Polymerase, again from the name, I have always told you initially that you must understand the terminology, okay? Ligase means ligating, joining. Polymerases, it will undergo polymerization, okay? Polymerization means the joining of the DNA or the bases, okay? It helps in the polymerization reactions. So it can be DNA polymerases, it can be RNA polymerases. Now, a very important enzyme, that is reverse transcriptase. So reverse, this is a general term. What about transcriptase? Transcriptase is something related to transcription process. So what happens in the transcription process? Here, from the DNA, mRNA is produced, okay? So from DNA to RNA. But here the name, I have said it as reverse transcriptase. So what does this signify? This means that this type of enzyme will do the reverse action. That means from RNA it will form the cDNA, okay? So that is complementary DNA. Now, what about the restriction endonuclease? We have already talked about the restriction endonuclease, correct? So it has certain specific sequences where they will cut the DNA, okay? So now, what about I'll talk about a very important term, that is the restriction modification system. This is very important in case of the bacteria, correct? Restriction enzymes are mostly found from the organisms, bacteria. Now there's a modification system also. Restriction is due to the fact that the enzymes, they restrict the foreign DNA. That means they cleave off the foreign DNA, okay? Now, what about modification system? Now you understand the fact that if a particular organism is synthesizing this type of molecules which is cleaving the DNA, then DNA, we know it is a universal thing, correct? So for every organism it's same, correct? We have the 4 base pairs. Now, so it can also cleave its own DNA. So how it is protected? It is protected by the modification system. Here, what happens right after the process of transcription, then right after the process of replication, actually methyl groups are added to its host DNA. So because of this, it is differentiated properly. So the enzyme, the restriction enzyme, do not cleave its own DNA, correct? Now, for the restriction enzymes, we must be very much aware of a particular terminology, which is palindromic sequence. That means these are the sequences which, if you read from right to left and from left to right, those are the same, okay? So suppose we have talked about the sequence of EcoR1. So the sequence of EcoR1, if you read from left to right, it is like GAATTC. Now if we talk about the same sequence in the next strand, it will be CTTAAG. So it is palindromic sequence, okay? Now, if we talk about this restriction modification system, this restriction enzymes, it can cleave the DNA, correct? Now this cleaving of this DNA, can be of 2 different types. First, due to this cleaving, different types of cleaving, it can form sticky ends. So what do we mean by this sticky ends? Sticky end means that the cleaving is such that on one strand there will be some overhanging, okay? Certain overhanging will be there. So it is sticky because it can stick to the other strand. What about the other one? Cohesive ends, that means the other one is the blunt end cuts, where the sequence is cut in such a way that there is no overhanging. So it will be sticky or the blunt end cut. So these are the 2 types of important cuts. So I'll just remind you once that please keep posting your questions. Please invite your friends and please subscribe to our channel at Toppr, correct? So please subscribe to our YouTube channel and please post questions. We'll be discussing about your questions. So we have talked about the sticky and the cohesive ends of restriction modification system. Now let's talk about a very important thing, which is gel electrophoresis. Okay, so what is gel electrophoresis? This is a very simple technique by which we have to visualize the DNA molecules. Okay, whatever DNA molecule, we know that these DNA molecules, they are very, very small. Now how can we visualize them? So this is a technique where we actually use to visualize this. So I'll just take up if any questions has come. We'll check for your doubts. So I've got a question which says, I want to understand the process of RNA interference, how it happens, and this has been posted by Mayank. Mayank, I'll be coming to that topic very soon. Please wait for that. Okay, so I'll cover that very soon. Now we are talking about the gel electrophoresis. Okay, so this process of gel electrophoresis, has a basic rule that it separates based on the size. under the influence of an electric field. So we have an agarose gel. So there, these DNA samples, they are loaded, okay? Loaded means just they are applied over there and they are allowed to run in an electric field. Now you know that the DNA molecules, they are negatively charged because they are negatively charged now. So they will move towards the positive end. So because based on the size, they will be separated. So you understand, if the size is smaller, then it can move easily. It can pass through the pores of the gel easily. And if the size is larger, they won't pass easily, correct? So this is the basis of the gel electrophoresis separation. The separation is not enough because we have to visualize the separated DNA. How do we visualize them? We visualize them by a process of staining, and the staining is done by ethidium bromide. Now I'll just point it out that this is an important area where questions are being asked. So one important question which has been asked frequently is that certain types of DNA are given. For example, the supercoiled, the linear and cleaved. So if I ask you that if I have given three types of samples, that is supercoiled DNA, the linear type of DNA and the cleaved. So, and I am running them in a certain gel at a particular voltage. So which one will be the fastest one to travel down along the gel? So what do you think the answer should be? The answer will be the supercoiled one will be the fastest, because you understand it is supercoiled, it is the smallest, it can easily pass through the pores. Whereas compared to that, if we compare about the linear and the cleaved, mostly the linear will be the faster one than the cleaved, because you understand if it is cleaved, it may have some overhangings. This may hinder to the easy movement about the gel electrophoresis process. Okay. So we have talked about certain techniques. We have talked about the gel electrophoresis, we have talked about various enzymes. So let's now talk about if any, let's check if you have some doubts about here. Okay, so Falak has asked, can you explain please about insertional inactivation? I'm sure, Falak, I will be explaining this in a while. Just be patient, I'm coming to that topic very soon. So let's talk about the cloning vectors, okay? Cloning vectors, we have already talked about that. These are the things which will help in carrying the DNA from the desired organism to a specific cells or the host, correct? So these are the vectors. Simple definition. One of the very important vectors, the first artificial cloning vector. This is an important question which is asked in examinations. Which is the first artificial cloning vector? That is, please remember, it is PBR322, okay? This is the first artificial cloning vector. So this was actually constructed by Bolivier. Now, we are talking about cloning vector. Now, everything cannot be a cloning vector. All the plasmids cannot be a cloning vector because of the facts we are going to talk about now. Now, what are the facts? Now, cloning vector, it has to have some features. What are they? First, they have to have the origin of replication. That means a sequence from which the replication process will be initiated. Now, what is important? You must be thinking. Now, this origin of replication is dependent on the specificity of the sequences. Some sequences are there which has got high specificity, because of which the initiation of replication is very fast. So, origin sequence is very important. Next important thing, very importantly, is the selectable markers. Okay. Now, suppose you have been able to transform. Now, how will you select which are the cells which have been actually transformed, which have received your desired DNA, which have not? So for this case, what we do is that the vector, it contains certain resistant genes. What are they? They are for example, the PBR322, it has got ampicillin and tetracycline resistant genes. Now these resistant genes, actually they help in selecting. For example, the fact that the cloning sites, this is the third part or the third thing which is very important for a cloning vector. Now this cloning sites are the areas where we are going to put our desired DNA. Now just see how clever and how intelligent we are. We have put this cloning sites inside this ampicillin or any antibiotic resistant gene sequence. Now what will happen? Just think, if a particular insert has gone inside this ampicillin resistant gene, then what will happen? It will result in the destruction of this ampicillin resistant gene. So if we culture this type of cells in an ampicillin containing plate, then it won't grow in those plates because it will die, correct? Now, similarly, we use the most general criteria is we use two types of selectable markers so that we can easily select or screen out the particular transformants. Okay. Now we'll be talking about the question which has been asked, that we'll be talking about the insertional inactivation. Now you understand. First of all, I'll try to make you understand why insertional inactivation. Already we have a process like selectable markers is fine. Why insertional inactivation? Because of the process that ampicillin and tetracycline resistant genes, if we follow this type of technique, then there are high chances that we select a wrong colony. Okay, we have to do plating, and there there's probability that we will select the wrong colony. So what we have done in insertional inactivation is that we use the enzyme beta-galactosidase enzyme sequence. So this beta-galactosidase enzyme sequence is incorporated. Now the cloning site is put inside this beta-galactosidase enzyme sequence. Now you understand, if there is no insert, now the property of the beta-galactosidase enzyme is that if we put a chromogenic substrate, that means a color giving substrate, it will give a rise to colonies which are blue in color, because the enzyme is active, it can convert it. Okay. So what will be done? The chromogenic substrate will be formed, so it will give blue color if there is no insert, because the beta-galactosidase enzyme sequence is intact. Now if we talk about if there is insert, so what will happen? The beta-galactosidase enzyme sequence, it is destroyed, it does not code for the proper proteins. Then what will happen? Will we get color? We won't. So from there we can easily select out the colonies or the actual transformant. I hope you have understood this insertional inactivation. This is very important. Now another important thing we'll talk about is the competent host. Okay. Yes, Javed, you have asked a question. I'm waiting for the topic process of recombinant DNA. I'm coming to that very soon. Now we'll be talking about the competent host. Now the host in which we are going to give or put the DNA, that should be ready. So this process is known as competent host, or we have to actually make the host competent. Correct? Now what are the various ways which we can do that? First of all is the calcium mediated form, which is one of the commonest form. Here what happens is calcium ions, they actually get attached to the DNA and they help in settling the DNA sequences or DNA strands over the surface of the cells. Now we give a heat shock, about 42 degrees centigrade heat shock for about 2 to 3 seconds. You understand, you must be aware that the cellular membrane, it is made up of lipid bilayers. Okay, so if we give a heat shock, there are pores which are formed. Due to the pores, it easily goes inside the cells, and since it's a shock, again it reforms back its own structure, original structure. So you understand, this is a very easy method through which we can put the DNA. There are other advanced methods where we use micro injections. Here micro injections are used to directly insert the DNA into the plants and animal cells. Also we use electrophoresis. This is here we actually create pores on the surface, which is mostly calcium chloride mediated, and also very important type of invention, which is the gene gun. Okay, here we bombard the cells which is covered, which is coated with the DNAs. Okay, so here it is bombarded. So mostly we use gold foils, correct? Gold particles which is covered with the selected DNA. Now gene gun is this method. Now we'll talk about the question we have got. We'll talk about the process of rDNA technology. Okay, so first what we should do, we have got a desired DNA, desired characteristics from a particular cell. So what is the basic step we should do? We have to isolate that. So we isolate that, correct? We lyse the cells, we isolate the products which is present inside the cells. Now there are contaminants, because there is proteins, there is RNA, there is DNA, there are various other things inside the cell. Now we require the DNA specifically. So we use proteases, ribonucleases, because of which these will cleave the proteins and the RNAs. So these are unwanted material for us, correct? And during this process, it is actually precipitated with chilled ethanol. Now you must please remember that this is one of the areas where it is asked that what is the chemical which is used for precipitation of this DNA molecules. It is chilled ethanol. Okay, now you have isolated. Now what will you do? You'll be cutting the DNA at specific locations. Okay, you'll be using the restriction endonucleases, correct? Now restriction endonucleases are used to cut the DNA molecules. After cutting, it is joined with our own with the specific DNA molecules or the vectors, okay, for example the plasmids. So after joining, what we have got the chimeric or the recombinant DNA. Now here, this recombinant DNA, we follow a very important technique. Again, I'll point it out that this is the technique which is very important, PCR, polymerase chain reaction. This was formulated by Mullis. Okay, this is a very important discovery because of which we have been able to do so much in biotechnology. PCR, polymerase chain reaction, it has 3 basic steps. First is denaturation. So what we do over here is we try to denature the double strands. We heat that the DNA strands at about 94 to 96 degrees centigrade. What will happen? The hydrogen bonds will be broken. 2 different strands are formed, correct? Now we use primers, RNA primers. We use 2 different types of primers specific for a particular sequence which we are desiring to amplify, correct? Got it clear? So we use 2 primers because there are 2 strands. One will be a forward primer, other one will be a backward primer, okay, reverse primer. So we use 2 different primers. Now the very important thing over here is we tone down the temperature over here. It is between 40 to 60 degrees Celsius. Why? Because if we keep that temperature there, then the hydrogen bonds cannot be formed. But we keep this type of temperature so as to avoid unwanted type of hybridizations. Okay, so here annealing is happening. So RNA primers have stuck there. Then the next step, which is extension, we use a very important enzyme which is known as stacked DNA polymerase. It is isolated from Thermus aquaticus. So it is one of the common questions which is asked. Yes, I am talking about the stacked DNA polymerase. I've got a question, importance of stacked DNA polymerase in this process. Mahinder, you have asked this question. So stacked DNA polymerase, this is very important because this is the enzyme, you know, this actually helps in the process of polymerization. Now you must be asking that there are various other polymerases, but why do we use stacked DNA polymerase? Now I have told you that this is isolated from a bacteria which is called Thermus aquaticus. This bacteria, these are found in thermal vents. So you understand this bacteria is live in thermal vents which has got a temperature of more than 100 degrees centigrade. So these enzymes, which are proteins which are very specific to temperature, they are active even at such higher temperatures. So you understand why do we use this type of enzymes? Because we'll be doing this extension process at a temperature of about 72 degrees centigrade. So here these enzymes, they remain stable, and this is one of the most stable enzymes, and also because of the fact that its efficiency of polymerization is very good. So these are the proper reasons because of which we are using stacked DNA polymerase, correct? Please note this cycle is being carried out for about 30 cycles. Now you may ask why not more than 30 cycles. We do not actually go beyond 30 cycles because the capability of the enzyme, it deteriorates after 30 cycles. So best product we can get till 30 cycles. Okay. So here we have got the amplified DNA. Then we move on with the preparation and insertion of the recombinant DNA into the host cells. Correct. So we have talked about the PCR, which was invented by Mullis. Okay. So now let's talk about the bioreactors. So we have got the cells, correct? Bioreactors जो cells हमें मिल गया, correct? So वो cells को हम क्या कर सकते हैं? What we should do is सिर्फ cells से काम नहीं आएगा. Okay, we have to amplify those cells, or we have to actually bring out the products. Okay, we have to bring out the products. So इसको कैसे कर सकते हैं? We use bioreactors. Okay, bioreactors हम use करते हैं. इसमें क्या करते हैं? 2 type of bioreactors हो सकता है. One is simple and the other one is sparged. So basically the difference between ये simple और sparged के बीच में जो difference is sparged bioreactors have bubbles of air which is sparging in between the reaction process. And what we do, हम क्या करते हैं इस process में, bioreactors में, actually we do the fermentation process. Okay, so यहाँ पे जो cells transform हो गया, उनको transform करने के बाद इनको fermentation process किया जाता है, and we actually use bioreactors जहाँ पे we try to maintain the conditions. Okay, conditions maintain करते हैं. अब fermentation process 2 type में हो सकता है, continuous and batch. Batch में क्या होता है? Like we do not change the nutrients. जब प्रोसेस चल रहा है फर्मेंटेशन के, तब हम न्यूट्रिएंट्स और माइक्रोऑर्गेनिज्म से जो प्रोडक्ट बन रहे हैं इनको हम निकालते नहीं है. वी डू नॉट चेंज इट, वी वेट फॉर द प्रोडक्ट्स टू डू. ओके, सो द प्रोसेस गोज ऑन देन. वेयरएज इन कंटिन्यूअस व्हाट हैपेंस, द न्यूट्रिएंट्स जो है वो हम रिप्लेस करते रहते हैं कंटिन्यूअसली. सो दिस इज द बेसिक डिफरेंस. Okay, so as of now we have talked about the main areas where we have covered the main principles. Okay, so let's talk about important applications. अब कुछ applications की बात करते हैं. ये बायोटेक्नोलॉजी जो हम प्रोसेस यूज करते हैं, फर्स्ट एंड वेरी कॉमन एग्जाम में जो पूछा जाता है वो है बीटी कॉटन. ओके, आई एम श्योर आप यू मस्ट हैव हर्ड अबाउट इट. ओके, सो आई हैव गॉट अ क्वेश्चन ओवर हियर. So this has been put forward by Aparna. Aparna, you have asked how to distinguish plasmid DNA and chromosomal DNA. Okay, so the basic point about this is plasmid DNA, you know, these are circular double-stranded DNA and they are capable of proliferating or replication. Self-replication is possible for the plasmid DNA. Okay, whereas the chromosomal DNA, they are not circular. They are mostly not circular. They are mostly there in the form of networks. Okay, and we can also talk about the size of the DNA. Like plasmid DNA, they are much smaller in size. Now if you ask about that, how can we differentiate it? It's a very simple process. We can actually do centrifugation in the presence of certain chemicals. Okay, so we can use actually phenol and ethanol. We can use these solutions where if we centrifuge them based on the molecular weight, we can actually separate these type of chromosomal and the plasmid DNA. I hope you have understood this. So we were talking about the applications, correct? So first we will talk about the BT cotton. Okay, is this, in order to talk about this, we'll talk about an organism which is Bacillus thuringiensis. Here this bacillus, this organism actually, it codes for an enzyme, codes for a toxin which is known as BT toxin. This is coded by the gene which is named as cry gene. Okay, okay, so Mahindra Sharma, you have asked about the elution process. Okay, so elution, yeah, I can shortly explain about the elution process. Once you have separated the DNA, correct? Once you have separated the DNA in the, where we have separated in the electrophoresis technique, correct? Now I've told you about that we use certain enzyme, certain labels or dyes. Okay, ethidium bromide. Now in order to visualize, we have to use a UV lamp. Now in the UV lamp we can cut that particular piece of the portion where we have a desired DNA. We can cut that, and we have in the market, we have available kits to extract these type of DNA from the gels. So from there we extract it into a solution, into a microfuge. You know, microfuge, they are very small tubes which can be centrifuged. Okay, they are known as microfuge. So we use actually that small part of the DNA, small part of the gel which contains our desired DNA. We microfuge, we centrifuge it with using the elution kit, and from there we can actually extract the DNA in our solution. Okay, so I hope you have understood this. Now we were talking about the BT cotton. So here the cry gene, it codes for the toxin BT toxin, and the property of this toxin is that it can actually kill various insects. Okay, how? This toxin actually create pores under alkaline conditions. Now you see our intelligence, how human beings have used this. We have done this biotechnological process, we have done this rDNA technique. Okay, we have talked about the rDNA process. We have incorporated this cry gene into the plants. So what happens? These cells, when the plant cells, they are being eaten up by these type of insects, what happens? Due to the alkaline pH of their stomach, it gets converted into the active protein, which is actually the toxins. Now these toxins actually bind to their gut and it creates pores. Pores bana dete, correct? Now when it creates pores, you understand, it actually results into the death of that particular insect. Insect धीरे धीरे मर जाते हैं इसके वजह से. पर अभी now the important question is, तो फिर ये क्राइजीन इस ऑर्गेनिज्म को क्यों हार्म नहीं करते, करेक्ट? ऑर्गेनिज्म को हार्म इसलिए नहीं करते क्योंकि ऑर्गेनिज्म के अंदर the gene which is being produced, or the protein which is produced, it is produced in an inactive form which get activated in the alkaline conditions. Now we have used this as because the gut of the insects, they are alkaline in nature. So in the alkaline nature, what happens? It is converted into the active form and it dies. So you understand what about the BT cotton? Now we'll talk about, we had got a question about RNA interference. Okay, it was put forward by Fire and Mello, correct? So this is a very important and interesting technique where the main target, you should understand about the target. So the target was to silence the mRNA. Why? Because from the mRNA we have the proteins. Now what we have done is that we have put the genes for particular, if we try to silence a particular mRNA, we put both the sense and the antisense genes into that organism, correct? What will happen now, there's a particular enzyme which is known as dicer. Okay, so because we have put both the RNAs, it will form double strand. Now dicer enzyme, it cleaves into small siRNAs, small interfering RNAs. Now these are known as interfering, correct? Now these siRNAs, they form complex with the ribonucleases, and it forms RISC, RNA induced silencing complexes. Now these complexes actually unwind. Once these complexes are formed, we have seen that it is double stranded, it unwinds and it gets activated and actually kills the mRNA, desired mRNA. So similarly, we have actually been able to process or make human insulin by the same method. It was the structure was given first by Sanger, and San actually synthesized human insulin for the first time. Okay, for the last few minutes, we'll talk about some of the important questions or doubts which you have put forward. So the first, the question which has been asked by Jaya is how to remember numerical values and names of fishes, etc. of this lesson. Okay, so yeah, this is a question, this important question, because I've already mentioned that biology involves too many terminologies. It's quite difficult to do that, but you can use some jargons. Okay, so you can relate, you have to relate those organisms with the name of the phylum, with the name of their characteristic features. You can name them, and in this way you can actually remember those. So now let's talk about the next question. We have asked about, Abhijit has asked about transformants. We have talked about the transformants, and next, okay, so we have also talked about the insertional inactivation as asked by Falak. Okay, please explain about artificial DNA. Okay, so artificial DNA, yes. So we can explain about artificial DNA. So what's artificial DNA? Like we have talked about the plasmid which has been made, correct? It was an artificial plasmid which was constructed because it's not a part of any organism. Now you understand, if we try to construct those type of vectors, we need to satisfy the conditions that it must have a ORI, it must have some certain selectable markers, and it has some certain the cloning sites. So we can actually create those sequences, and artificial DNA sequences has been created, and they actually help like as very good vectors. Okay, so now we have also talked about Mahendra's question, which is about TAC polymerase. We have talked about that. Okay, so yes, Javed have asked, we'll take up the last question, which is Javed have asked about that full form of the BT. Okay, so yes, so BT cotton, this has actually come from the name Bacillus thuringiensis. Okay, Bacillus B and T thuringiensis. So from here the name BT toxin has come. So everyone, I hope you have enjoyed this session. Please do not forget to subscribe to our YouTube channel. Please follow us on YouTube. Invite your friends also on Instagram and Facebook. If you have doubts, please put forward, and we'll be trying to give answers for your questions. Surely, I hope you have enjoyed. Thank you.